DNA is a long molecule that contains our unique genetic code. It is found in the nucleus of a cell and has a double helix structure. DNA is made up of nucleotides. Nucleotides consist of a phosphate, sugar, and nitrogenous base. The nitrogenous bases are adenine, thymine, guanine, and cytosine. For the double helix to form, the nucleotides join to each other due to complementary base pairing, where adenine binds with thymine and guanine binds with cytosine. Protein synthesis is the process by which amino acids are linearly arranged into proteins through the involvement of ribosomal RNA, transfer RNA, messenger RNA, and various enzymes. The process has two main components: transcription and translation. DNA transcription is a process that involves using the DNA strands as a template to make the messenger RNA strand, which is processed during the translation phase. Let's take a closer look at transcription. During initiation, which is the first stage, RNA polymerase binds to DNA at a specific sequence of nucleotides called the promoter. Here, the transcription of the gene begins. The RNA polymerase makes its way along the gene and unwinds the DNA. As the DNA is unwound by the RNA polymerase, only one strand acts as a template for the RNA synthesis. During the second stage, known as elongation, free ribonucleotide triphosphates floating in the cytoplasm pair up with their complementary bases exposed on the unwound DNA strand. As this is an RNA strand, the nitrogenous base, uracil, replaces thymine and pairs with adenine. The RNA polymerase links to the nucleotides to the 3' end of the growing messenger RNA molecule. RNA polymerase zips DNA back up as it goes, only leaving around 10 to 20 bases exposed at once. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, DNA strand, and the messenger RNA transcript unattach from each other. The newly formed messenger RNA strand leaves the nucleus through a nuclear pore and travels into the cytoplasm. Translation involves decoding a messenger RNA strand and using its information to build a polypeptide, which is a chain of amino acids. Let's see how this is performed. The nitrogenous bases are grouped into 3-letter codes called codons. Most codons code for specific amino acids, apart from 4 special codons: 1 which codes for translation to start, and 3 which code for translation to stop. For translation to begin, the messenger RNA strand must bind to a ribosome, which is found in the cytoplasm. The ribosomal RNA associates with a set of proteins to form ribosomes, which act as the docking bay for translation to take place. Translation begins when the messenger RNA strand binds to the small ribosomal subunit just upstream of the start codon. After the initiator transfer RNA molecule carrying the first amino acid enters the ribosome and binds to its complementary base, the large ribosomal subunit binds to form the translation complex and initiation is complete. From there, each amino acid is brought to the ribosome by specific transfer RNA molecules. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. As the transfer RNA molecules enter the ribosome, the amino acids form a chemical bond with each other to form the polypeptide chain. The transfer RNA molecules detach from their amino acids and exit the ribosome, making room for the next charged transfer RNA molecule. The polypeptide chain is complete when the stop codon is reached. This polypeptide chain is now a protein. And as we know, proteins are what make up our body. This is where we can observe a link between the products of translation and an individual's phenotype. Let's use an example to demonstrate this further. When the gene was being read and used as a template in the transcription phase, it was coding for a specific trait or characteristic. During translation, this code was used to produce a polypeptide chain, which will make this trait or characteristic apparent in an individual. For example, let's say the gene being read was coding for eye colour. The sequence being read and copied on the gene due to transcription was instructions to make the protein melanin during translation. For the purpose of this example, the gene coded for a protein that only makes a little bit of melanin. Therefore, the individual's phenotype for eye colour will be blue.